Hello, everybody. Welcome to the Labflow workshop, the experiment number 10 for 1406, which is carbohydrates. Let's start to say what are carbohydrates. I will give you a quick overview of carbohydrates, and then we will go over the report to make sure you know how to complete the report. So we said, what are carbohydrates? They are polyhydroxy aldehydes or polyhydroxide ketones, or compounds that yield to polyhydroxide aldehydes or polyhydroxide ketone when they suffer hydrolysis. This is a lot of words, so let's go over here this formula. We have here the drawing of glucose. We said that glucose is a carbohydrate. So when we see this drawing this way, we have to make sure that we know what that means. Glucose, first of all, has 6 carbons. 1, 2, 3, 4, 5, and 6. The carbon number 1, as you see here, has an aldehyde group. What is an aldehyde group? That you will have to learn. This is the aldehyde group, which means the carbon has a double bond with oxygen, then has to have a hydrogen because it's the carbon number 1, and then the rest is bond to another carbon. In general, we can say R. So it has to have an aldehyde, and then all of the other carbons, let's do it in another color. Carbon has a valence 4. 1, 2, 3, 4. So each carbon is going to be bond, let's do it this way, to one hydroxide. This group, OH, is the hydroxide group. And then I'm trying to draw the glucose. So every intersection of the line means that there is a carbon. And then in glucose, the carbon, this is carbon 1, carbon 2, carbon 3. Here is the OH. So the carbon is bound to an OH and an H. To an OH and an H. To an OH and an H. Sometimes you will see this written like that, CH2OH, the last carbon, which is this part. Very good. So when we read the definition again of carbohydrate and we say polyhydroxide aldehydes, well, now it's much easy. Poly means there are many. There are many hydroxides. 1, 2, 3, 4, 5. Polyhydroxide aldehydes. Aldehydes is because we have the aldehyde group. And the classification, as you see here, this is the drawing of glucose. Let's look at fructose. Fructose is also a carbohydrate, but look at the difference. Look at the group that is a difference. This one we said is an aldehyde, right? The carbon 1. And look at the carbon 2. It's a ketone because the carbon is bound to with a double bond, is bound to oxygen, but the other two bonds are to carbon. So this is a polyhydroxide ketone because the ketone group, and polyhydroxide because there are many hydroxide. Now that we are familiar with the structure of the carbohydrate, we will know that there is a classification of the carbohydrates. They are classified as monosaccharides when it's just one molecule. For example, glucose, as we just draw here, this one, or fructose, those are monosaccharides. Then we can have disaccharides. For example, sucrose or lactose. Why they are called disaccharides? Because they are two monosaccharides that are joined, that are bound together. We will see that. We will see later how's the name of the bond. And then we see polysaccharides. Ooh, look how many in this drawing are joined together. For example, starch or cellulose, there are thousands of monosaccharides linked together. In this experiment, you can check with our instructor. Your main objective is to perform different tests to distinguish between different types of carbohydrates. So we are going to perform 4 tests mainly. The first test is the Benedict's test. The Benedict tests are used to identify simple reducing sugars. Reducing sugars have an aldehyde that can reduce other compounds. Let's look at here. This is sucrose. It's a disaccharide, and this is a non-reducing sugar. So the Benedict tests will tell you if you are in the presence of a reducing sugar or a non-reducing sugar. A sugar is classified as a reducing sugar only if it has an open chain form with an aldehyde, as we just seen the glucose, or a free hemiacetal group. We will go over what is a hemiacetal group. It's an hemiacetal is an alcohol and an ether attached to the same carbon. And the fourth bonding position is occupied by a hydrogen. So let's look in here. Here we have we are looking for, in order to be a reducing sugar, we have to have an aldehyde or an hemiacetal group. The structure here, it says, lacks an hemiacetal acetal functional group. That means the sucrose is non-reducing. How do we know? By looking at this structure that is not a reducing sugar. We have to look at the carbon 1. Look at the description. We said that an hemiacetal is an alcohol and ether attached to the same carbon. What is an alcohol? We have to have an alcohol. For example, this is ethanol. When are you in the presence of an alcohol? When you have an OH group. And when do you have an ether? When you have an oxygen bond to 2 carbons. For example, this one. But in this case, hemiacetal group, it says an alcohol and ether attached to the same carbon. So let's look, for example, let's look in this alpha carbon, this one of lactose. Let's pay attention to this carbon. This carbon, we are talking about lactose, is bound to an alcohol, right? This bond going down, and the other bond is an ether. So this alpha carbon is an hemiacetal. That means it is a reducing sugar. The same thing happens here. This carbon is bound to an alcohol and an ether. This is another hemiacetal. So lactose and maltose are reducing sugar. But what happens with sucrose? Why it's not? Because when we look at this carbon, they are locked. They are locked. The carbon that is bound to the ether is bound to 2 other carbons. It doesn't have the alcohol, none of them. That's why sucrose is not a reducing sugar. Of course, if we hydrolyze that, yes, if we produce hydrolysis of the sucrose, then it will create 2 reducing sugars, because sucrose, when we do the reaction of hydrolysis, it will produce, it's formed by glucose and fructose. So the glycosidic bond is broken. And how do we do the hydrolysis? The hydrolysis is done by adding strong acids or enzymes. So let's review a little bit the first test that we are talking about, that is the Benedict test. The Benedict test is, we said we are going to identify simple reducing sugars. Now, how about polysaccharides? For example, amylase or amylopectin, they actually have a hemiacetal group on only one terminal end. But what happened? They're non-reducing substances. If you have a polysaccharide, it will give the test, the Benedict test, negative. Why is that? Because there is only one reducing group over 2,000 or 5,000 monosaccharides units. So the test, the Benedict test, is going to be negative. Very good. So all the complex carbohydrates, like starch, do not react with the Benedict's solution. So let's share now the page of the experiment. So we said that the first test that we are going to do is the Benedict's test. Very good. And here is showing what is an hemiacetal, what we just talked. This carbon is bound to an alcohol and an ether. Then the glucose is going to be a reducing sugar. Here is a drawing of the equilibrium. Most of the time the sugar is cyclic, but in this case, for this test, we don't need to get into that. So here is showing this is the reducing end because this carbon is bound to the OH and the ether. And here, no, this carbon doesn't have the OH. So then it's the non-reducing end. So here is the reaction of the Benedict's test. What is really happening? You need to learn this reaction. So you will have in your tube, let's say, for example, glucose. Glucose will give you a positive test. What you need to know in this experiment is what is the signal, what is the change in color when you do the Benedict test. Well, you are going to have your tube and you are going to watch the video. Let's say you have glucose, right? And you are going to add a solution of copper +2. Here. And if you look at the reaction, there is a redox reaction. That's why it's called reducing sugar. The copper +2 converts into copper oxide 1, and it changes the color. It gives you a brick red precipitate. This is red. And if you look at the oxidation state of this compound, if you do you will see that copper is copper +1. And here it was copper +2. That means the oxidation state from the copper went from 2 to 1. It was copper is reduced, and this changed the color from blue. We add a blue solution to red. But actually, the color of the product depends on the concentration, how much reducing sugar you have in your tube. It can be either green for low concentrations, or yellow, or orange, or the red brick. Remember that to see this positive, you need to place it for a few minutes in a water bath. So what is important from this reaction? What do you have to know? If you have a positive test, it will look, as we said, brick red because the reduction occurred, or depending on the concentration, you can have a green, or you can have a yellow or an orange. This is the change in color. And if you have a negative reaction, you will see a clear and blue color of copper to sulfate. So if you see the change from blue to red, then there is a reduction of the Benedict's reactant. Now, every time you have a reduction, you have to have an oxidation. Who is oxidizing in this reaction? Here, at the beginning, we said that this is an aldehyde group, which is a carbon that has a double bond to oxygen and one to hydrogen. So we have an aldehyde. And what happens here? What group is that? This is a carboxylic acid. The carbon has the double bond, but instead of a hydrogen, it has another oxygen. So when we say about oxidation, how are you going to figure out? Because the carbon has one more oxygen, and that is one way for organic compounds to figure it out if there is an oxidation or if there is a reaction. If the carbon gained oxygens or lost hydrogens, which is our case, here there was a hydrogen and there is no hydrogen now, then there is an oxidation. But if we go the other way around, from the acid to the aldehyde, it's a reduction because this carbon is bound to two oxygens and here is bound to one. So losing oxygen or gaining hydrogen means there is a reduction. But for our purposes, this a reducing sugar will reduce the copper sulfate of the Benedict's test and is going to oxidize itself into a carboxylic acid. But one more thing, the Benedict solution also contains sodium carbonate to keep the solution basic and sodium citrate to complex the copper-2 ions and prevent other reductions. Because we have a basic solution, they are drawing the carboxylic acid group as this one. If you are in the presence of an acid medium, instead of writing like that, the carboxylate, this is the carboxylate, you write the group, the carboxylic acid, OH. This is going to protonate, and this is a carboxylic acid. But the carboxylic acid will lose the proton, will lose this H if we are in a basic solution. Very good. So I think for the Benedict test, we've seen a lot. What is the next step? The next test. Here they are talking about that not all disaccharides contain a reducing end. We just seen that. And the next test is the iodine test. Why are we doing the iodine test? Just to check if a sample contains starch. If a sample contains starch, it will react with the iodine solution to form a blue-black solution. If a sample does not contain starch, the solution will have the color of the iodine, that is yellow-brown. And the test requires the complex carbohydrates to succeed. What are complex carbohydrates? They are polysaccharides that are also called complex carbohydrates. And starches is one of the most common polysaccharides. And starches are formed mainly from a mixture of amylose and amylopectin. And the amylose is about 15%. They form like a little coil. So the iodine will be inside, will get inside and produce that blue-black color. The amylopectin is like 80%, is also a linear chain, but it's a branched molecule that is made of thousands of glucose units. Very good. So the second test we said is the iodine test, just to test starch. And starch forms a dark blue color complex with iodine. So the straight chain component of the starch, that is amylose, will give you a blue color, and the branch component, amylopectin, will give you a purple color. So we said that in the presence of iodine, we said that the amylose molecules form helixes. And inside of those helixes, the iodine molecules will enter, and that's why we see that black-blue color. And then we are going to perform the third test, that is the fermentation test. Fermentations, you heard more about it, is the process in which the carbohydrates are broken into an alcohol by a specific substance. For example, yeast will make most of the monosaccharides and disaccharides undergo fermentation, and we will obtain ethanol and carbon dioxide. Let's see if there is a reaction here. Yes. Okay. So all we have, this is the formula of the ethanol. It's just 2 carbons. It's an alcohol, has the OH group. So when you are, the process of fermentation will give you the alcohol and a gas, carbon dioxide. So how are we going to know that the fermentation is positive, that we have a positive test? We will see bubbles. In this experiment, we will use lactose and galactose. And those two will produce negative results. The glucose will undergo fermentation. You are familiar probably with the maltose that we have in barley or other grains. And what is the process of the beer? First, there is a hydrolyzation using some yeast or enzymes, and then you obtain glucose. And then the glucose is used in brewing a beverage and cereals. So first they hydrolyze the barley, and then what happens? Once you have the glucose, they undergo fermentation to obtain the ethanol and the carbon dioxide. But for us, the only objective in order to know if we are in the presence of fermentation is to look for bubbles. And the last experiment is this one, the Seliwanov test. What is that test for? It will show if we have ketosis or if we have aldoses. Basically, if we are in the presence of aldoses, as you know, contain aldehydes, as we saw at the beginning, and ketosis has ketones. And this test is just to distinguish those. All you have to remember is when is a positive test, when is a negative test. If you have a ketone, or a ketose, you will see that this test is positive when you see the color red, bright red. And if you don't have a ketose, you have an aldose, then you will see a light pink color, and it will come not that quickly. So basically, this test immediately will tell you, if it reacts quickly, that you have a ketose. For example, fructose, as we saw at the beginning. And then I think we can go now. Here is the procedure. We can share the page of the report. The report shouldn't be that complicated, but let's try to, it's in reality, it's very simple, because all you are doing, let's pay attention, is these 4 experiments. Benedict test to check for reducing sugar, Seliwanov test to check for ketosis and aldoses, the fermentation test, and the polysaccharide iodine test. All we have to remember, this is data that I put it there, but you will look at your data and you will write it there probably, or if it comes like that, I believe you have to pass it from there is a data page, and you will have to transfer that information there. So let's look at my sample. Everybody, so you will compare all these 4 different reactions with different sugars and different carbohydrates, and depending on the color that you obtain, you will decide if it's a positive reaction or if it's a negative reaction, and then they will give you an unknown. So this is data that since you are not in the lab, this data is given. So the first thing, all you have to do is, for example, let's look at here. You will have to complete that. They will ask you, Benedict test is to check if you have a reducing sugar. First of all, is the water a sugar? No. So then we can say neither, right? Okay, that is not that clear, doesn't matter. And then what happened with the water? Are we gonna see something? No, we are not talking about a sugar. So this is neither. Do we have, is the water fermenting? Is the water going to give us ethanol and carbon dioxide? No. Is the iodine test positive with water? No. We need a starch in order to be positive. So we complete all that. Let's look at glucose. What did we say about glucose? Is glucose. Let's look at the data before. We know now, because we went over before, why glucose is a reducing sugar. But let's look at here. You will have to base on the colors. Glucose, it says brown red. Brown red, that means the reaction is what? Positive. The reaction is positive, that means we have a reducing sugar because it changed the color. If the reaction is negative, it will stay clear blue. Fructose, yes, fructose is a reducing sugar, so then it's positive. What happened with sucrose? We said at the beginning of the workshop, we show why sucrose is a non-reducing sugar. And here in the test, we see the color of the reagent, of the Benedict's reagent, that doesn't change. So then it's a negative reaction. Lactose, it's a positive reaction because it changed the color. And the starch, blue, is a negative reaction. So this is basically what you will have to do in this report. Let's look at the ketosis and aldoses. What do we say? Just to check if you have an aldose or if you have a ketose, right? And what did we say? For example, for glucose, we know it's an aldose, right? Because when we look at the formula in carbon 1, we have an aldehyde group. So we know that glucose is an aldose. Does it match with the Seliwanov test? Yes, because when we see light pink, that means that we have an aldose. That is the color of the aldose. This test is just to distinguish if we have an aldose or a ketose. What happens if we see a red? Oh, we have a ketose. So here we have a red, and based on the color, you will write on the other part. And let's look at the fermentation test. Water, obviously, nothing. If you see bubbles, we said that the test is positive. So when here it says tan and bubbles, the glucose is giving you a positive, right? And then this one is giving you a positive. Here is giving you a positive. It says none, obviously negative. And that's the way you are going to complete that report. Let's look at polysaccharides. Iodine test. What do we know? That is positive with starch. With starch, it will give you a blue-black color. Which one is the only one that is giving me a blue-black color? Well, we know it's the starch. This is positive. And the others are all negative. So basically, this is all you have to do in this part. Here it says, hydrolysis of disaccharides and polysaccharides. Here is given the data. And again, basically, what you have to do in this part is look at the color. When do you have a positive reaction? If you see a blue, is it positive? Is it positive? No. If you have a brown blue, yes. And that's what you are going to continue. And in the last part of the experiment, you will have to, again, based on the color, you have your unknown, and based on each test, if it's positive or negative, this is mine, for example, that I said that I have glucose, but depends which unknown each person has. And basically, this is simple based on the colors, since you're not doing the experiment. And here I'm concluding this workshop, and I would like to thank everybody that will listen and or everybody that is here now. And I will stay in case you have questions. And thank you again.